right, 2 of chromosome number 1 and 2 of chromosome number 2, and when I get to the end, I still have 2 of the same chromosome 1 and 2 of the same chromosome 2, right? Asexual reproduction or binary fission results in identical clones, right? In meiosis, though, right, I don't have any chromosomes at the end that look like chromosomes that I started with at the beginning, right? Because they switched some information for one, right? That leads to genetic variation, right? This chromosome might have gotten new alleles of that gene that it didn't have before. This chromosome also, right, might have new alleles of genes that it didn't have before. The other thing, right, that leads to genetic diversity is that when these homologous chromosomes line up, right, I drew the white one going this way and then the white chromosome number 2 going that way, but it doesn't necessarily happen that way, right? It could have been, right, that the black one was here and the white one was here, and then down here, right, the black one was on this side and the white one was on this side, right? And so when these chromosomes split, right, it's just as possible that this cell ended up with one version of chromosome 1 and the other version of chromosome 2, and vice versa. Right? So the homologous chromosomes, right, since the chromosomes themselves are going separate directions, right, we don't know, right, which of these chromosomes it's going to get. Right? It might get, right, the same chromosomes, right, that match each other that came from the original, or it might get different chromosomes than it ever had. Plus those chromosomes are different because they traded information. Right? So 2 ways there that we increase genetic diversity, right, by kind of mixing and matching chromosomes into new cells, and also by those chromosomes flip-flopping a little bit of their information between each other. So practice that because it will definitely be on the exam in basically exactly that form. Okay, what are Mendel's 2 laws of inheritance? One of them sounds like segregation is one of them. Yeah, what's the other one? Independent assortment. Very good. What does segregation mean?
Organizing by.
All right. What does independent assortment mean?
Organizing by some same category.
Yeah. So at mitosis, right? We're talking about mitosis again. Right, segregation, right, means that the genes for the chromosomes, right, or the genes exist on the chromosomes, and the chromosomes are splitting apart from each other. Just like you said, right, they're segregating from each other, right, and so those genes, right, get pulled apart, right, and then they come back together, right. Independent assortment, right, means that we don't really know, right, which one's going to go which way because it's totally independent, right. Just like we have over there, that's also independent assortment. Right, we don't know which chromosome is going to go which way and which is going to go the other way. And over here, right, we don't necessarily know, right, which way something's going to go. Let's see here. Laws of probability. That'd be a good one for us to go through. All right, so let's take our example. I'm going to erase this. Anybody who wants a picture of it, take a picture of it. I think we have one more guest that wants to tell you about what he's running for. Yeah, sure.
Hello everyone, my name is Alex. I'm running for Chief Justice with the council right now. As you may know, Abraham was here and in his campaign. Also working in police. I'll be running for Chief Justice. So basically what I do as Chief Justice is run and be an ex officio advisor to the board of trustees or the CEOs of the campus basically. So yeah, you guys so much. I'm taking much of your time. Yeah, please vote for me. And Abraham as well, because he's already, so thanks so much.
I think I told you all I was actually president of the Nutritional Science Student Association when I was in grad school. I both hated that experience and learned a lot from it all at the same time. Let's see here. Okay, we're going to do some probability. So let's take a cross. Right, we already know that if we have right, one parent that has, right, is heterozygous for a particular allele, right, and we cross that with another one that's maybe homozygous recessive for that same allele, right, then we could pretty easily just make a Punnett square, right, and we get a heterozygous, 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 and in this case, homozygous, right? And so the proportion of offspring that we get that are going to be heterozygous, just like one of the parents, is going to be 75%, right? And the proportion, right, that's going to be homozygous recessive is 25%. You're right. There we go, 50-50. Right? That's easy enough, right? However, we know, right, that there are a lot more genes, and we know that those genes, right, assort independently. So again, right, the genes, right, they segregate on their chromosomes, right, and each gene is kind of on its own, right, for doing its own thing. Right, so we could consider a cross that was much more complicated. So that's very simple. We just make a Punnett square and we're done. Right? But what if we've got 3 genes to consider? So for example, what if we take a parent that's heterozygous for everything, and we make that with a parent. Go. Something like that, right? So now we have 3 different genes. So it becomes a little bit more complicated. Yes. They are. They're both lowercase. No. They're both. No, one of them is way bigger than the other one. Draw it again. P is not a good letter to use for this example. Whatever. Here, I'll draw this one really big. There we go. Right. So now we have 3 genes to consider. Right. But right, the odds of getting one or the other allele is 50%. Right. You can only get right 1 of 2 alleles from this one, only 1 of 2 alleles from this one, right? And so we can use probability to determine, right, all the potential crosses. So what are all the potentials, right, for this particular cross? There are 12 of them. So for example, right, we could get, right, a dominant and a dominant. Right, with a dominant and a recessive, right, and also a dominant and a recessive. Right, so that's one particular cross. We can also get 2 dominants, right, heterozygous again, right, but we can also, right, get a recessive from both of those. That's a possibility. So we could have homozygous recessive. We can also have the same dominant there, right? But recessive for Y is also a possibility, so those could both be recessive. And we could again have heterozygous for R, right? Or the last possibility is homozygous dominance, right? And then it's recessive for both of those. Okay? So that's 4 potential crosses. Instead of being homozygous dominant here, we could be heterozygous. So we could be heterozygous there, right? And then again with the same combinations here. Or, all right, so there's 4 more possibilities. Right? Or, right, it could be homozygous recessive for that first one. And so we can also have that, again with all the same combinations. Okay, so those are our 12 possibilities. Right, for that particular cross. Right? And now we just have to look at what the question's asking us. Right? So if I asked you what are the odds, right, or what proportion of offspring will have all three recessive traits, right? Well, that's only one possibility. That's this one. If I ask you, right, which one, or what proportion of offspring will have exactly two recessive traits, well, that would be this one, this one, this one, this one. Missing one? Two recessive traits. Yeah. All right, so there are four of them. Yeah.
Would you have to consider that there's essentially two sets of the heterozygous as well? Like, there's, you do. There's you'd have two sets. Because you get the lowercase for either side. So that bit where there'd be two sets. That
Let me see. Why don't you write it out and then we'll review later. What if I asked you, right, what is the proportion of offspring that have at least two recessive traits, like the example that we did in class before? Well, at least two would be all the ones that have exactly two, plus the one that has three, so now we have five potential combinations. Right? Or I could ask you, right, what proportion of offspring will only have one recessive trait? Right, in which case we're only looking at this one, or this one, or this one, or this one, or this one. Right, so all things I could ask you, right? What proportion have one recessive trait? What proportion have two recessive traits? What proportion have three recessive traits? What proportion have at least two recessive traits? At least one recessive trait, right? So the question will vary. Right? But if you can put out all the possible combinations, then just look at what I'm asking you for. Right? And let's do the one that is exactly two recessive traits, right? So the green starred ones. Well, then we can do a Punnett square. So, for example, right, with the peas, both heterozygous. So when we look at this cross, right, we know that homozygous recessive is going to be 1/4. Homozygous dominant is also going to be 1/4. Heterozygous, I think, is going to be 2/4. Or 1/2. So, right, for this first one, right, it has the homozygous recessive, right, which is a 1/4 chance of getting that. Just put 1/4 for that one. And this one also, right, 1/4 for having that proportion. This one, right, is heterozygous, which was a 2/4 possibility. And this one was homozygous dominant, which again was 1/4 possibility. And then we do our next one, right, for y's. That's big Y, little y, and then little y, little y. Right, so in this particular case, right, the odds of getting homozygous recessive were 2/4. And the odds of getting heterozygous also 2/4. Right? So this one's heterozygous, so that's the 2/4. Well, they're all 2/4, so this one is 2. This one is also, and so is this one. Right? And then our last possibility would be R's. Right, the R's, it was heterozygous and homozygous recessive, just like it was for the P's, so the proportions will be the same. Right, Right, and so to get homozygous recessive was a 1 out of 4 possibility. Right, to get homozygous, and again, was 1 out of 4. For this one, right, also homozygous, so 1 out of 4. And for this last one, the heterozygous was a 2 out of 4 chance. So, now that I have all of the potential possibilities, I've chosen just the ones that I care about for this particular problem, and I have calculated the proportion, right, of offspring that would have each of those 3 alleles independently, because independent assortment after all. Right, now I can just multiply those together. Okay, so this is 1 times 2 times 1 is 2 out of 4 times. How do I do fraction math again? I forgot. Right? I feel like my math is wrong here. Oh yeah. Times 4, 16 times 4. I've done something wrong here.
The R should have the same like double recessive, homozygous recessive.
You think I missed some combinations?
So like here, you have the double recessive R and 1 4, 2 4. Like do punch squares the same.
Okay, I just messed up mine. You're right, so. E, P, Y, Y, R, R, yeah, you're right. It's all halves.
So all the R's should be 2 No, you have to first. No, you have to. Right, you have to first. It will be 1 out of 4. So the Y's and the R's should be the same grade. Both of the phenotypes, the Y and the R's, are 2 out of 4 because and 4. Because like for the Y's and the R's, you know, when you have the Y experiments, I have it on like
No, 64.
Six. This one too.
you roll like 1. So it's 1, so 2 over 4 times 2 over 4. Yeah.
1 equals 4 times 2 is 8. 64. This one is also, let's see here. Right?
I want
4, and the last one is heterozygous R. There we go. So now 8 and 4 is 12. 8 is 20. 24.
64 equals 12 out of 32 equals 6 out of 16. Come on. There we go. 3/8. Final answer. All right. A little bit of math difficulty there. You can see how it's easy to get mixed up. Right? All the more reason to practice. Right? Because even sitting here with some scribbled notes on note cards, I still managed to get some of my numbers mixed up. Did I get the right answer in the end? Yeah. So there we go. And so now what we have, right, final answer here, 3/8, right, is the proportion of offspring that would have exactly 2, right, recessive traits, right, because I only did the ones with the green stars. If we had done, right, at least 2 recessive traits, right, then we would have thrown in this last variant here because that's got at least 2, right, it has 3, but it has at least 2, right, and so our proportion would have gone up just a little bit. But that's how you can take that concept of, yeah.
Well, it was 24 over 64, and I just simplified it to 3/8. Right, so I took each one independently. All right, so 8 out of 64 plus 4 out of 64, that's 12. Plus another 8 is 20, plus 4 is 24, so I had 24 out of 64, and then I just simplified the 3/8. Right? Okay, well, that's what I mean. Do we understand the process?
Yes.
Right, me doing it on the spot on the board, obviously I'm getting my math wrong, but we understand the process, yes? All right, let's move on. There will be a problem like that on the exam,
Is it, Professor?
so practice it a couple times.
Is it only going to be 3?
I don't know, there might be 4 or 5. You understand the process though, right? Yeah, it's not a matter of do you understand this specifically. Yeah, I'll probably keep it to 3 or 4 max, right? Because with 4 genes, right, then the number of possibilities increases a lot. Right? So, yeah, probably be 3. Yeah, that's what everyone was saying too. I put the wrong proportion on there. Yes, you're correct. Yes. All right, this one's actually a 1. Let's go ahead and fix it. It's actually 23.
Okay, that's 4 out.
Oh yeah, that makes that 4. Now it's 20. Now it's 5 out of 16. There we go. All right, moving on. Let's see here. Okay, so complete dominance. Right, remember that complete dominance is where the heterozygous phenotype is the same as the homozygous dominant phenotype. Right? That means as long as you have one allele that produces the protein, that protein can create the entire phenotype, and so that is complete dominance. Right? The recessive allele, right, plays no role. It doesn't add anything in at all. Right? It doesn't contribute anything, and so it has completely been dominated. Right? The example we saw in lecture, right, pigmentation of a flower. Right, if it the allele for making the what was it, a purple pigment, I think. Right, if it has that allele, it makes the purple pigment. If it doesn't have that allele, it doesn't make the enzyme, and so it just doesn't have purple pigment. Right, and so it's a white flower. Right. If it's a heterozygote, right, it's got one allele to make that enzyme and one allele not to make the enzyme. But since it at least has one allele to make the enzyme, it can still make the purple pigment, and so you still get purple flowers. Right. Only in the case where you are homozygous recessive do you not have any ability to make that enzyme, and so you have no ability to make that pigment, and that's the only time you get that white flower. Incomplete dominance, right, is when both alleles, right, are going to contribute something. Right? An example we saw that was with Tay-Sachs disease. Tay-Sachs disease was one in which an enzyme for processing lipids is not produced. Right? In this case, right, a person with that is homozygous recessive, right, doesn't make that enzyme at all. they can't process that lipid at all, and so they have Tay-Sachs disease. Right? If they are homozygous dominant, right, they have two good alleles, so they make two good enzymes, and so they have no problem processing all of that lipid. They don't have the disease. But if they're heterozygous, right, they have one allele for making the enzyme and one allele that doesn't make the enzyme, and that one allele that makes the good enzyme, right, means they can process some lipid, but not as much, right, because they have lower enzyme levels. And because that enzyme isn't sufficient to process all of the lipid, the same as if they had two good alleles, right, we call that incomplete dominance, right? Because they do have lower enzyme levels, they just don't have zero enzyme levels. Another case of incomplete dominance, or no, sorry, then codominance, right, is where you have two different alleles that exert different effects, but none of them dominates over the other. So one good example was with blood type. Right? In blood type, you can be type A or you can be type B. Right? Type A and type B, right? Those are different things. Right? You can have one, you can have the other, you can have both at the same time. Right? But one doesn't cover up the other. Right? They have different functions. Right? And so they're codominant because they both exist in equal proportions and they don't affect each other in any way. Other terminology we have for this, right? So multiple alleles can show up as either pleiotropy or epistasis. Multiple alleles just means that, that multiple genes even, right, can function in ways that depend on each other. Right? So in pleiotropy, right, you have a couple of genes, right, because very often the phenotype is due to more than one gene. Right? That's the idea here, is that it's not just one gene, one phenotype. Right? Sometimes a phenotype requires more than one gene for its complete expression. So pleiotropy is when one gene is dependent on another. The example we saw was with the Labrador Retriever. Right? So you have one gene that is responsible for what color pigment it makes in the first place, right? And then you have a second gene that determines whether or not that pigment actually makes it into the hair. Right? So it doesn't really matter what color pigment you make if you don't have the enzyme to put that pigment into the hair. And it doesn't matter if you have the pigment to put into the hair, right, if the enzyme that makes the pigment, right, doesn't make that pigment in the first place because there's nothing to put in the hair. So that's pleiotropy, right, when you have two genes, right, for two different processes, but those processes are dependent on each other for the final phenotype. This is similar to epistasis, right? Epistasis is also, I just said that wrong. That's what epistasis is. That's what epistasis is. Right? That's the one where you have two genes dependent on each other. Pleiotropy, right, is where one gene has multiple effects. Right? So, for example, in cystic fibrosis, what's wrong is a sodium transporter. Right? A sodium transporter leads to really thick mucus in the lungs, which is why people with cystic fibrosis have lots of lung congestion. But that's not the only place that you see that sodium transporter. So they also have symptoms associated with that faulty sodium transporter in other parts of their body. That's what pleiotropy is, right? Pleiotropy is where one gene, right, can have an effect on multiple different symptoms because it affects multiple different parts of the body. Epistasis is where you have two genes that are reliant on each other for the final phenotype. And then polygenic inheritance, right, is where multiple genes are involved in some phenotype, but they're not necessarily related to each other. So for example, in human height, right, your ability to absorb nutrients, right, it might be one factor that leads to how tall you are. Right? The amount of growth hormone that you produce might be another factor, right, that leads to how tall you are. Those don't depend on each other. Right? Your nutrient absorption and your growth hormone production don't depend on each other, so it's not epistasis because one doesn't affect the other, right? But it is still two genes that affect the final phenotype. All right, what shall we say with this one? All right, does everybody remember Morgan's experiments with the fruit flies? Let's do that one real quick. What did we learn from the fruit fly experiment? What was, like, if you had to say one thing, right, just a couple of words, what would you say was the result of the fruit fly experiments? It is relative to mutation, right? Because having white eyes versus red eyes is a mutation, right? But specifically, what did we learn about that mutation? Where is it located? always in the DNA. So Morgan and his fruit flies gave us information about specifically X-linked mutations. Another little scribbled out note card. Let's see if I can get this one a little bit better. All right, so remember when we do mutations, we typically identify the gene based on the mutation. Right? So the mutation is having a white eye, so we label it with a W. Right? And so if you have a female fruit fly, right, that has red eyes, that means she's W+, right? W because the mutant is white, but if you're W+, that means you're not the mutant, right? And so this would be red eyes, and this gene happens to exist on the X chromosome. So we get 2 of them, right, because this female has 2 X chromosomes. If we cross her, right, with a mutant fly, so we just write W, no plus, that means he has the white eyes, so he's the mutant. Right, and then his other chromosome is Y, right, so I'm just going to put a Y there, right, because he can't have another allele for that gene because this gene only exists on the X chromosome. Right? So if we cross those together, we're going to get, right, W+, that's the red-eyed female. Right, and then we have the white-eyed male with his white chromosome. And so the offspring of the F1 generation is going to look like this, right? So F1, remember, is the filial generation. This is the parental generation up here, right? And what we have here, right, are 50% of our offspring are female, right, with red eyes, just like the parents, right? And then the other 50% of the offspring are male, right? And they, right, also have red eyes, right, because they got, right, the non-mutant allele there, right, and then a Y chromosome. Yeah, right, so we have some heterozygotes, right, but they do have one good allele, so they have red eyes. And then we over here, right, our males, right, are by they're obligate, right, to be homozygous because they only have one allele possible because they only have one X chromosome possible. If we breed those together, right, then our parental generation is going to be, right, either it's going to be a heterozygote female with red eyes, right, and then all of the males have that phenotype. They're also heterozygotes, right, by virtue of the fact that they only have one allele in the first place, and the other chromosome is a Y chromosome. Right, and so what we're going to get out of this and if you look at this, right, a quarter of them are homozygous for the red eye, right, the females are. Another quarter of them, right, also have red eyes, but they're heterozygous. Right, these will also have red eyes, right, but because they only have one Y chromosome, right, and then here, right, we have the ones with the white eyes, right, so these are red, 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 and white. Right? And that's what Morgan observed, right, was at any time, right, we're getting the same proportions, right, but any time we got one that had white eyes, it was always a male, right, and that helps to bring evidence to the fact, right, that some things are X-linked, right, which means they're more likely to show up in males because males only need one recessive copy to exhibit that trait. A heterozygous female needs both copies of that recessive gene or that mutant, right, in order to have that because she has 2 possibilities of that allele, but the male only has 1. So that's what Morgan's experiment with the fruit flies, right, provided evidence for, right, was that some things are X-linked or exist on the X chromosome. Let's see here, some examples of sex-linked disorders include color blindness, Duchenne muscular dystrophy, as well as hemophilia. So all 3 of those are things that are X-linked disorders, just meaning that the allele for that is on the Y chromosome or is on the X chromosome, which means the males only have to get one bad copy in order to have that. Let's see here. Then we have the closely related concept. Right, so this is, right, X-linked, right, which means it's linked to the X chromosome. We also just have this concept of linkage of genes. right? Not X-linkage, but just linkage. And linkage means genes that are really close to each other, so that when, right, during meiosis, you have this chromosome, which I'll shade in, it replicates itself, shaded in still. Then we have the other copy of that chromosome, and it replicates itself. If we take this little band right here, and we put it over here instead, right, and the band that was there is over here instead, right, there might be 3 genes on there. If those 3 genes are really close together, right, then when we trade a little bit of this chromosome with that chromosome, those 3 genes will travel together, right, and that means that they're linked, right? So linkage just means the genes are really, really, really close together on the chromosome, and therefore they're more likely, right, to trade with each other. Versus a gene that's way down here, right? This gene is probably not linked with these 3 genes, right? Because the odds of getting this section all the way down to here and trading that whole thing, very, very, very low. They only trade a little bit of material back and forth. So the further apart the genes are, right, the more they behave as if they were on totally separate chromosomes in the first place. The closer they are, right, the more likely they are to travel together. That distance is called a map unit. Right? So the distance, right, between these 3 genes, right, is going to be a few map units. A map unit indicates every 1 map unit is a 1% chance of recombination, and that's what that is. That's recombination, right? Because we now have new combinations of genes we didn't have before. Right? So if they're only, right, 1 map unit apart, right, that means that there is a 1% chance, right, that they will not recombine, or a very good chance that they will recombine. Let's see here. What else did we talk about in this chapter? Oh yeah. Sometimes chromosomes exhibit non-disjunction. Right? So that's an alteration of the chromosome number right here. Right? Non-disjunction just means that the chromosomes don't separate from each other. Right? They stay stuck to each other. And so when those sister chromatids move one way or the other, right? If these 2 sister chromatids separated and one went to one cell and one went to the other cell, right? These should also separate with one going to one cell and one to the other, but if they have non-disjunction, they stay stuck together, right? Then both of these, right, might also go into this cell, and just that one went into this cell, and so now right, we have that shaded one, plus the one that experienced non-disjunction, and then over here we have the other shaded one. And so that leads to an aneuploidy, right? Non-disjunction, right, means that this one ended up with 3 copies of that chromosome, right, because those chromatids didn't separate like they were supposed to, and so this one only got the 1 copy because it didn't get its other copy. That could lead to something like trisomy 21, right? So if this was chromosome 21, right, we see this new cell has 3 copies of 21, and this cell only has 1 copy of 21, and trisomy 21 is known as Down syndrome. That's non-disjunction. There are other things that can happen too during crossing over. Right? So during crossing over, right, an analogous portion should get traded, right? But sometimes that doesn't happen. Right? You could get a duplication, for example, right, where you get twice as much as you're supposed to. Right? You could get a deletion, right, where in that trading of material, right, maybe, well, that here as well. Right? Maybe instead of flip-flopping these 2, right, this one accidentally kept its part. So here, right, we have the original, and then it got a piece from that one. Well, this little piece comes over here and adds in there. Right, and so now this chromosome has 2 copies of that particular region because it forgot to trade its copy over. Right, it kept its piece of the chromosome, and it got the analogous piece of the chromosome, right, from the other one, and it added it to it. And so now we have a duplication over here and a deletion over here. So mistakes can happen, right, during crossing over, where you get, it could also flip-flop upside down. Right? So as this piece, right, is supposed to move over here, right, it might actually invert, right, and now it's in there in the reverse order. I think that's another possibility. So several different possibilities during crossing over that could lead to something being wrong. Let's see here, and then this last little piece here, Right, organelle genes, that's referring to mitochondria and chloroplasts. Right? Those are the two things that we saw with endosymbiont theory, where they're thought to have arisen from prokaryotes earlier, so they have their own genetic information. So mitochondria don't show that Mendelian inheritance because it's not part of the nucleus, and it doesn't involve any of this movement of chromosomes. And then imprinted genes, that was the example of that was epigenetics. So, for example, if your grandmother had a particular diet, right, with lots of methyl groups in it, and those methyl groups attached to the DNA that can silence portions of the DNA. And when that DNA, right, if it's in a gamete, right, gets passed on to offspring, then that DNA will still be silenced in the offspring because the silencing happened in a gamete. If the silencing happened in a somatic cell, like a skin cell, that wouldn't make any difference because I don't pass my skin cells onto my offspring. Right? But if the silencing happened in my sperm, right, I do pass my sperm on to the next generation. Right? So some of my genetic material, some of the chromosomes in my sperm, right, were silenced by overmethylation. Right? Then when those chromosomes ended up in the egg that got fertilized, right, then they would still be silenced, and so my kid, right, would have some silenced regions of its DNA. All right, moving along. Should we take a break? What time is it anyway? Yeah, let's take a little bit of a break. It's been a little while, so let's do maybe 10 minutes. Take a break, and then we'll finish off this review and do